You know that since we started late, we are falling behind. And on future lectures, we have to unite some lectures. Some of the, for example, cementum and alveolar bone, and periodontal ligament and the gingiva, and so on. Today we'll deal with the dental pulp. You know that we discussed dentin, and you know that dentin and the dental pulp form such one complex because the pulp tissue is located in the chamber, rigid chamber, which is formed from dentin. Dental pulp is the soft, loose connective tissue. We studied such mineralized tissue, such as enamel, you know, the most mineralized tissue in the body. Then the dentin, less mineralized, but more than bone and the cementum. And today we discuss this is a dental pulp, which is very important, but it is soft, loose connective tissue, which is located in the central portion of each tooth. Uh, let let's see this picture. Yes, you see the dental pulp. It has crown. It has crown and radicular parts, crown and radicular pulps. You know that pulp is delicate, specialized connective tissue which contains thin walled blood vessels, nerves, nerve endings, which are enclosed in within dentin. You know that what is characteristic feature for connective tissue? That is the abundance of extracellular matrix. And the dental pulp, it has extracellular matrix, gel-like extracellular matrix, in which the fibers, a lot of types of cells are embedded in this extracellular matrix. Then each pulp opens into the tissue surrounding the tooth, the dentinum, and it is these are the tissue, periodontal ligament, and the gingiva and alveolar bone, which fixes the tooth in its socket. And through the apex of the root canal, the blood vessels and the nerve endings enter into the pulp. And here you can see the pulp. It's diagrammatically, and you know that topographically a distinction is made between the coronal and radicular pulp, and the shape of the coronal cavity roughly corresponds to the reduced and simplified form of the dental crown, with the vault widened under the cusp tips and the incisal edges. And the portion of the pulp in these widened areas are known as pulp horns. And here you can see these pulp horns. All the teeth have a pulp roof, which is formed from the dentin that overlies it occasionally. And multi-rooted teeth also have a pulp cavity floor, and thereby the pulp, it has the six surfaces, which is the mesial, distal, occlusal, buccal, lingual, and floor of the pulp. Uh, and in general, the root canals follow the course of the roots. You can see these are root canals. And you know that these root canals, they can form such accessory canals. You know that here in the apex of the root there is the foramen, apical foramen at the root apex, and each root has at least one main canal, you can see, and the apical foramen, and many have two, and small accessory here and here and here canals branch from the main canal and have their own foramina, and they are found most commonly in the apical third of the root. And here you can see this is the accessory canal. In a multi-rooted teeth, some small vascular canals enter the pulp chamber from the bone between the roots. And here you can see this canal, and it is about 13% a single bifurcation canal extends from the pulp chamber to the intraradicular region. And here, this is about 23% a lateral canal extends from the coronal third of the major root canal and to the furcation region, and about 10% have both lateral and furcation canals. And here it is a section of tooth apex which illustrates this accessory canal. Here you can see accessory canal, and this is the main apical canal, and here this is radicular pulp root which branches and gives such accessory canals. You know that this soft, loose connective tissue in the pulp, this connective tissue, it has its central zone, and it has its peripheral zone, and they are observed in both coronal and radicular pulps. You know that the volume of pulp also differs in different teeth. For example, molars pulps are 4 times larger than those in the incisors. And here you can see this is the frontal view, and this is the lateral view, and you can see the from first to the eighth third molar, and you can pulps which has sometimes in incisors and canine each has one canal, and here you can see the 2 canals and 3 canals in molars. Then what about the and here you can see this diagram shows the age changes in the S. You know that the dental pulp is such soft and very dynamic tissue, The composition of any connective tissue varies during the development with aging. And it changes when the tissue is injured or challenged with the bacterial toxins, and different cell types may be activated, migrate to the tissue, or develop from stem cells. Due to the condition, the age, this dental pulp composition is permanently changed. It is very dynamic tissue, and there may be the degradation of extracellular matrix, and larger molecules are built up from smaller components, and sometimes the small components, small molecules may be broken down products of the larger molecules, and this cause this dynamic composition of the dental pulp. You know that and here you can see how abundant these cell types are here. There are some cell types, and we discuss it a bit later. Profusely vascularized, profusely innervated tissue, and here you can see the less number of cells, specific cells, and the less number of fibers. You know that. What is connective tissue? It is extracellular matrix, which is composed of fibers. The main type is collagen fibers, and also the cells, different types of cells. And here you can see that the chamber of dental pulp chamber is reduced, diminished, and here this is age changing, and this is attrition also affects pulp hole with appearance of dead tracks, you can see, and sclerotic dentin. And you know that the secondary dentin, which is throughout whole life, is laid down, it decreases the dental pulp chamber. And here you can see, and also sclerotic dentin. This is age changes. This is the cause of different trauma or attrition or abrasion, or and this is the middle age. It is the young stage after some attrition, middle age, and it is old age. Now, then next. Yes. What about the composition of the dental pulp? Dental pulp contains abundant extracellular matrix. The basic scaffold of pulp tissue is produced by collagen fibrils, collagen fibers, and thin fiber bundles. And there is the main collagen type is type 1 collagen, which is ubiquitous type of collagen. And there is about 60% of such collagen type. And also you can find here the collagen type 3, which is about 40%. The proportion of type 1 collagen in dried pulp is 25%, or in premolars, and 30% in molars. The pulp tissue of the anterior teeth contains even more collagen fibers. In the root area, the collagen fibers are packed more tightly into fiber bundles, such that the proportion is higher than in coronal pulp. The total quantity of collagen fibers remains constant in the coronal pulp after the age of 20 years. More collagen is generally found in humans than in most experimental animals. The type 3 collagen is the proportion of type 3 collagen is high, as we know, about 40% of the total collagen content. This high proportion is responsible for the elasticity of pulp tissue. Though the individual collagen fiber is not elastic, a network of collagen fibers form a mesh that as a whole exhibits elastic characteristics. This particularly affects type 3 collagen because its fibrils are thinner than those of type 1 collagen. Type 3 collagen fibers are known as reticular fibers and are thinner than type 1 collagen fibers. Collagen fibers have been detected between odontoblasts by electron microscope. And here you can see these are collagen, this is ground substance and fibers, collagen fibers of the extracellular matrix. Then collagen type 5 and 6 also are found in the pulp. forming a dense meshwork of thin microfibrils throughout the pulpal connective tissue. And 5,4 collagen has been detected in the basement membrane of blood vessels in dental pulp. Uh, the compared with other dental tissue, the buildup and breakdown of collagen is relatively high in pulp. And you know that the main cellular component of the connective tissue is fiber cell called fibroblasts. And fibroblasts, you know that they are capable of producing and disintegrating collagen. And for example, when bacterial infection and inflammation are present, intensive collagen breakdown is caused by the degradation activity of fibroblasts and the bacteria producing collagenase themselves. Then the collagen synthesis of fibroblasts during the formation of tertiary dentin is increased. Here you can see this is the collagen fiber here indicated, and you can see these fusiform cells which are fibroblasts, and the synthesis of, during formation of tertiary dentin is increased, especially after administration of calcium hydroxide for the dentistry purposes. The most apical portion of the radicular pulp contains densely arranged collagen fibers and forms a transitional tissue to the highly fibrous periodontal ligament. And here also you can, this is the other method, Virchow method of staining, and you can see how these fibers are stained in black. And here you can see the normal dental pulp, which is represented by loose connective tissue, and you can see the plenty of collagen fibers and rare fibroblastic cells. And here you can see this is the special staining for glycosaminoglycans in blue. They are stained in blue color. And here you can see that these are collagen fibers, which are in periphery. They are arranged in such parallel rows. And here also you can see the collagen fibers, this blue appearance of collagen fibers. And here you can see this appearance of elastic fibers. Elastic fibers consist of thin microfibrils, which are known as oxytalin fibers and have woven elastin in between. And these elastic fibers are found together with blood vessels in pulp. Then, what about extracellular or ground substance? And you know that all the pulp cells and the fibrous network are embedded inside gelatinous ground substance that contains mainly water, about 75% water, and consists of proteoglycans and adhesive glycoproteins. And proteoglycans, you know, here we have, yes, proteoglycans, which are glycosaminoglycans. You know that they are attached to the protein core, and they are functionally diverse molecules. Some of them, for example, versican it is the large hydrated aggregate, and it has a molecule. And also you can see here the such proteoglycans such as fibronectin, and also there are four groups of cell adhesion molecules. They are general in this extracellular matrix, and the immunoglobulin selectin, the caterins and integrins. They are responsible for junctional complexes for cell-to-cell adhesion, and the selectins have a very special role in guiding the lipidases of leukocytes during inflammation. And here this shows this dark part on the periphery. It shows the proteoglycan versican, which is most intense in the sub-odontoblastic zone of coronal pulp, and here and here and here. And also you can find here the fibronectin, which is concentrated near the odontoblast layer and binds these cells to the extracellular matrix. As the extracellular matrix is abundant, these cells are adhered to this, and this fibronectin, versican, and this decorin, for example, binds to type 1 collagen, for example. Integrins, these are cell surface adhesion receptors. These are transmembrane proteins and are adhesion molecules. Overall, proteoglycans have a high viscosity and a filtration function, which presents a barrier against the spread of invading bacteria. Some bacteria, for example, streptococcus, produce hyaluronidase, which an enzyme that greatly reduces the viscosity of ground substance and favors to spread of bacteria. Then the extracellular matrix also contains such these adhesive glycoproteins such as fibronectin, and these are very significant in interaction of cells and the junctional complexes and interaction between the cell and the matrix. And this fibronectin is present in the odontoblastic layer, especially at the border between cell membrane and predentin. It is therefore concluded that fibronectin helps to create a tight seal in the location. Higher concentration of fibronectin are also found around the blood vessels, and generally speaking, increased amount of fibronectin are produced during wound healing and hence during the formation of reparative tertiary dentin. What is the next? And here you can see the zones of the cells. Pulp, you know that it is a loose, specialized connective tissue, and here you can see dentin. What is interesting about pulp and dentin? It is one complex, as we mentioned, and they are derived from dental papilla. I repeated it many times, and like to recall that there is tooth germ, which consists of three parts. It is enamel organ, which gives rise to the enamel. The second is dental papilla, and dental papilla, it has a mesenchymal origin, and it gives rise to dental pulp, and then the specialized and differentiated cells, which are odontoblasts, they give rise to dentin. They have a common origin, dentin and dental pulp. Thereby, it is one complex. And this they differ very much from each other. from one another because dental pulp is very soft, very loose connective tissue, and the dentin, it is mineralized connective tissue. And the dental pulp consists of cells, intracellular ground substance, fibers, nerves, blood vessels, and the lymph vessels. And we try to discuss each component in turn. The ground substance permeated by fibers provides structural support and support for the cells and the vessels in the dental pulp. And there are different tissue zones in pulp, and they can be distinguished. And the first layer near in the boundary with predentin is odontoblast layer. We briefly discussed odontoblast, but now we can discuss this layer. This layer is odontoblast layer, and each consists of differentiated cells. It is outermost layer, superficial layer of the dental pulp. Beneath the odontoblast layer lies Weil's cell pore zone of the cell-free zone or cell-poor zone, cell-free zone. This layer is not truly cell-free because under electron microscope it was revealed that they contain a lot of fibers, lot of processes, but this layer has not nuclei of cells, and it may be called nucleus-free layer. And there are a large number of nerve endings and cytoplasmic processes of other cells. And the nerves pass through the entire pulp, but there is a preferred distribution of nerves in pulp depending on the type of nerve fibers. Beneath this cell-free zone, there is cell-rich zone. This is called also Weil's cell-free zone. This zone each contains the typical pulpal cells and undifferentiated mesenchymal cells, which can act as reserve cells. These pulpal cells are mainly fibroblasts. You know that main component, the cellular component of connective tissue is fibroblasts, and various immune system cells, because immune system cells are also abundant in dental pulp, and they provide this immune response and mm, dysfunction for whole tooth. Then you know that pulp also is permeated by blood vessels, lymph vessels, and nerve endings. Now about odontoblasts, here we can see these are the cells, these are principal cells of the pulp. You know that pulp extracellular contains extracellular matrix. It contains fibrils and fiber bundles such as collagen, versicon, other elastin fibers, reticular fibers, and also it contains cells which are odontoblasts, fibroblasts, undifferentiated ectomesenchymal cells, macrophages, these are immune cells, and other immunocompetent cells. About odontoblasts, here you can see these are odontoblasts. They are differentiated from mesenchymal, ectomesenchymal cells. You can see that beneath this odontoblast layer, there is the cell-free zone, which contains the processes of different cell types. And after beneath it, it is the cell-rich zone. Odontoblasts, you know that odontoblasts are responsible for the formation of dentin. They are differentiated, we mentioned they differentiated from pulpal cells, from mesenchymal cells, And in the fully developed tooth, the odontoblasts continue to lay down secondary dentin. You know that these odontoblasts, they throughout life, they produce dentin throughout life, and this is the physiologic secondary dentin. They are columnar. These cells are columnar, actively secreting cells, and they has diameter 5 to 7 micrometer and high 40 or 50 micrometers, and resting odontoblasts are shorter and less polarized. What does it mean, polarized? They have a very junctional complexes, and these form their basal end and apical end. And you know that releasing the dentin takes place from apical end of the odontoblast, where cell processes are located in the tubules. Then there are a few organelles in the odontoblast process which instead contains abundant microfilaments and tubules that lie parallel to the long axis. And such in the tooth, which is subjected to a mild insult or such as dental caries or other trauma, odontoblast is able to respond to laying down tertiary reactionary dentin, which is essentially an accelerated deposition of secondary dentin. And you know that the odontoblast is post-mitotic cell and cannot divide. And this layer of odontoblast layer, and here you can see this layer is single layer of odontoblast, but this crowding of cells causes such appearance. And what is this appearance? This appearance is called pseudostratified. It is not stratified, it is a single layer of cells attached to the predentin surface, and coronal odontoblasts are, columnar, and they have this crowding causes that they have nuclei at different levels, and it seems that it is stratified, but it is a single layer of odontoblasts. And they this gives such false appearance of multiple layers and is called pseudo stratified layers. What you know that here you can see this is odontoblast layer, this is forming tooth pulp with its cells, and this is formation of predentin, and this is predentin, and as predentin is formed, pre-enamel begins formation. And here you can, from the other side, from opposite side, you can see amyloblasts. And here you can see the odontoblast layer. This is pulp odontoblast layer. And also you can notice that there are blood vessels and plexus of blood vessels in odontoblast layer, beneath the odontoblast, and also inside this layer. And you can see these blood vessels in this layer. The odontoblast layer becomes a flatter layer of cells, and the number of cells declines by apoptosis. And it has been estimated that half the odontoblast in the premolar will die in the during 4 years, and following the completion of root formation. Through the odontoblast layer, prime role is in dentinogenesis. It has other significant properties that help preserve the well-being of the pulp. It acts as selective barrier, reducing the speed with which toxins can reach the pulp, while at the same time allowing tissue fluid from the pulp to enter the circulate and perhaps circulate within the dentinal tubules. Odontoblasts are capable of producing pro-inflammatory mediators in response to bacterial toxins such as polysaccharides, and may produce cytokines such as interleukins, which then can activate and trigger immune response for macrophages and for other immune cells. Then it is such a suggestion that odontoblast could act as a sensory receptor, passing on information from the outer dentin to nerve fibers in the peripheral pulp. But there are gap junctions, and they also have the receptors for these gap junctions for potassium. And these channels may have a role not only in mineralization but also in the transduction of mechanical displacement of into electrical signals. And here you can see the junctional complexes. You know that they are very tightly adhere these cells to each other. And these junctional complexes are desmosomes. Desmosomes, these all macular adherence junctions, they have a clear intercellular component as well as intracellular system of anchoring. And you can see these are desmosomes, fibrils, and are largely responsible for mechanical union. These are two odontoblasts, and they have this desmosomal cell junction between two odontoblast bodies, and junctions that completely encircle the cells are not present here. And also there are other junctions such as tight junctions. Tight junctions, you know that these tight junctions are very important for polarity of these cells, and these tight junctions appear as a near fusion of opposing cell membranes and limit the permeability of the cell layer. The more tight junctions are there, the closer these cells are together, the lower the permeability is of these cells. These junctions do not completely encircle the odontoblast and so limit rather than eliminate permeability. And there they also adjust their polarities for apical and basal poles of these. And also these tight junctions, we may say that they also add to the mechanical integrity of the layer. Then also you can find here, you can see the gap junctions. This allows the movement of small molecules directly between adjacent cells. It is important in cell-to-cell communication and would presumably have a role in synchronizing the activity of all the odontoblasts in the layer. Then these cells, odontoblasts, during tooth development, the activity of the odontoblast is affected by many signaling molecules and growth factors, and they also contain specialized pattern recognition receptors for these growth factors, and these not all growth factors, but these can initiate an immune response by secreting chemokines that recruit immature dendritic cells and beta defensins that kill bacteria. The next layer we can discuss. The next layer, you know that this was the odontoblast layer. In the here you can see this is the odontoblast layer. The next layer is a cell-free zone, about 40 micrometer in width, and it is under odontoblastic layer in histologic sections, and you know that it is called the Wales zone, and it is also called cell-free zone, and also it is called cell-free or bipolar zone that follows the Wales zone. Here you can see, and blood vessels, you know, can pass through this zone, and there is the dense meshwork of mainly medulla-free nerve fibers which run through this Wales zone, and the processes of fibroblasts, which are linked together by desmosomes or tight junctions. This also can be found, and this also creates a close connections with the odontoblast, and the manifestation of this Wales zone is highly variable, and the next zone is cell-rich or bipolar zone, which has a lot of cells, and the main cell type is fibroblast. Um, and fibroblast can be found in a resting or active functioning state, and they are still undifferentiated replacement cells of pulp, and these fibroblasts are the most common cells found in dental pulp. And we can see here, you can see these fibroblasts and these fibroblasts, and they are called the stellate cells, and they form junctions between cells, between their communication and the junctional complexes on their processes of these cells. They are flat, spindle shaped, and their nucleus is large and oval. And here you can see they can be such fusiform, spindle shaped, or flattened cell, and the nucleus is oval, large and oval. They bear numerous, sometimes long cytoplasmic, you can see cytoplasmic processes that are in desmodontal contact with adjacent fibroblasts. Fibroblasts can be present in an active functional position, or passive stage, resting stage. They are numerous throughout the pulp except for the subodontoblastic layer, cell-free zone. While they are not present in the zone for the most part, their cell processes protrude into the zone, and fibroblasts are particularly abundant and tightly packed in this cell-rich zone, bipolar zone of the coronal pulp. And as elsewhere in the body, the fibroblasts produce collagen, from which the fiber or network of the pulp is built up. This provides a basic scaffold for its stable but also elastic, because of this special network of fibers, which supports all other cellular tissue of the dental pulp. They also produce the intercellular substance consisting of glycoproteins and glycosaminoglycans. Uh, the morphology of pulp fibroblasts is dependent on their particular function. Many actively producing fibroblasts are found in young pulp, and these have a well developed rough endoplasmic reticulum and suitable field Golgi apparatus. The nucleus lies in the side of the cytoplasm, and the cell membrane exhibits numerous irregularly branched cytoplasmic processes. And the resting fibroblasts, no, and the resting fibroblasts in older pulp are smaller, they are spindle shaped and have markedly fewer processes and contain a less pronounced endoplasmic reticulum and a less developed Golgi apparatus. And here you can see the fibroblast, you can see the unmyelinated nerve axon, and also you can see capillary, and here there is no collagen fibers. And the next type, which is very important now and very promising cell type, and the question of this is the dental pulp stem cells. These are abundant. There are undifferentiated mesenchymal cells. You can see this is the tissue culture, mesenchymal cells, and they are present in pulp primarily in the cell-rich zone, subodontoblastic zone, and in the vicinity of blood vessels. They can be confused with fibroblasts under light or electron microscope, and the characterization of this cell type remains ambiguous, and these cells, they are linked to each other, you can see by their processes. In the dental pulp, many of these cell types in the dental pulp, including the odontoblasts, defense cells, and possibly many fibroblasts, are terminally differentiated, and although able to respond to stimuli, uh, are unable to differentiate into another cell type. You know that there, what is stem cell? Stem cell is the undifferentiated cell which can divide, which can proliferate, and can differentiate into other cell type. What is the characteristic feature for stem cell? Stem cell is the population when it divides asymmetrically. It means that one cell, one daughter cell, stays to replenish its own population, and other cell, daughter cell, differentiate into other cell type. The more cell type the stem cell can differentiate into, the higher its potency, differentiation potency is. And thereby there are some different potential cells. For example, totipotent cells, which are blastomeres, and they can give whole embryo with extra embryonic placenta. These are blastomeres. They totipotency means potency for whole, for all. They can give whole embryo with its placenta. The second, but by differentiation and by growth, according to the growth of embryo, the cell potency is restricted, and the next potency level is pluripotent cell. When, for example, internal mast cells, which are or neural crest cells, which can give rise to about 200 cell types. They are pluripotent cells. After restriction, there are multipotent cells, and such cells are these dental pulp stem cells, which are very promising. This is very promising field in medicine, and this population is a multipotent cells such as hematopoietic stem cells, such as myeloid stem cells, lymphoid stem cells, which give rise to several cell types, and these cells, this population of cells in the dental pulp, it can produce chondrocytes, osteoblasts, and other cell types, adipogenic, they have ability, adipogenic ability, osteogenic ability, chondrogenic ability. They can also be made to differentiate into both neural and vascular endothelial cells. And these dental pulp cells, stem cells, they can be found in two regions of the dental pulp: peripherally beneath the odontoblast layer, and more centrally around the blood vessels. And they are multipotent cells and can give rise to several cell types. They have been cultured and used to replace nerve defects, and this field is a very, very promising field. And here you can see these when we said, if you remember, that tertiary dentin was produced by odontoblast-like cells. You know that odontoblasts are post-mitotic cells. They cannot divide, they cannot proliferate. But these ectomesenchymal cells, they can differentiate into the odontoblasts or odontoblast-like cells and give dentin. Then the next cell type here, let's see. Yes, there are, you know that there are a lot of immune cells of immune system in dental pulp, and these cells are defense cells, and these cells may be named such as granulocytes, which take part in, you know that there are two types of immune system in our body. The first is primitive primary defense line, which is innate immunity, and the white blood cells such as granulocytes, you know that some leukocytes, they have granules, and these granules are very important. All cells have granules, what they are azurophilic granules. Granulocytes, they have specific granules, and they contain in these granules the enzymes such as lysozyme, defensin, myeloperoxidase, for killing, bacteria killing mechanism, bacteria killing enzymes. And you know that there are three types of granulocytes, such as neutrophils, eosinophils, and basophils. And let's discuss these cells. This cell is macrophage, also macrophages, and the lymphocytes. And let's discuss macrophage. Macrophage, you know that it develops from a monocyte of the bone marrow, and it is derived from monocyte. Monocyte is derived in bone marrow from myeloid lineage of multipotent bone marrow stem cell. And macrophage via bloodstream enters, it is distributed via bloodstream, it leaves the bloodstream and then enters the site of inflammation or infection. And depending on prior contact with infections and stimulation by cytokines, they can take on different shapes or in different tissues. And their names varies. They can found in lungs, for example. There they are called dust cells. In skin, they are called Langerhans cells, and other. They are everywhere in connective tissue, and they are anti-inflammatory cells. They form bridge between innate immunity and acquired. Innate immunity is non-specific immunity, yes, and also there is acquired immunity, the higher level of immunity. And these cells are phagocytic cells. You know what is phagocytosis? They can recognize foreign particles such as invader bacteria or pollen or dust or fungi, and the phagocytose engulf this invader pathogen. Then they digest them, and after this they release some chemokines, some cytokines, and they can activate other cells, such as granulocytes, such as a lymphocyte. And here you can see that there are macrophages. These are large cells here you can see. And these cell types, these macrophages, this is special staining from macrophages. And here you can see this is electron micrograph of macrophage, and it has a very highly lobulated nucleus, abundant amount of cytoplasm. And these cells, they release cytokines as bactericidal enzymes, proteases, lipases, and growth factors. And uh, three types, only three types cells, such as macrophages, dendritic cells, and B lymphocytes, are antigen presenting cells. And let's say briefly what is antigen presentation. When they engulf this invader or pathogen and digest them, after this, in a special protein molecule, which is called the major histocompatibility complex, MHC complex, they display inside, they bound the remnants of this pathogen with this complex. They digest this pathogen, and some peptide, small peptide from 8 to 10 amino acids or remnant of this pathogen, they bind with MHC complex, which is major histocompatibility complex, and then display on their surface. And this process is called antigen presentation. And these macrophages, dendritic cells, and the B lymphocytes are professional antigen presenting cells. They After this, T lymphocytes recognize this antigen, and the T lymphocytes after this trigger the immune process against these invaders. Such macrophages, dendritic cells, and B lymphocytes are key players in acquired or specific immunity. you know that macrophages are identifiable by from their irregular enlargement of their cell surface area, and this facilitates phagocytosis. And you know that numerous lysosomes are visible in plasma, and the rough endoplasmic reticulum, Golgi apparatus, and they are very prominent in these macrophages. And these macrophages, which primarily occur close to blood vessels, have a rather elongated shape. And also, there are also connective tissue cells, which are called mast cells. Mast cells also are very important cells. They are granular cells. It is somehow obscured their origin, because they have a lot of commonality with the basophils. You know that there are granulocytes, and these granulocytes are neutrophils, eosinophils, and basophils. And what happens when there is infection? These cells, these granulocytes, after activation of macrophages by interleukins and other activating chemokines or interleukins, these cells, endothelial cells of blood vessels, they retract to each other, and such gaps or clefts appear between these endothelial cells. And these granulocytes, they can leave bloodstream and via diapedesis. Diapedesis means that they have amoeboid, they are spheroid, they are spherical in bloodstream, but in connective tissue they acquire such amoeboid form, shape, and they can move in the connective tissue, and they direct to the sites of infection or inflammation. And these neutrophils, eosinophils, and basophils, as you know, they contain specific granules. Thereby, they are called granulocytes. And the basophils are very like, they have a lot of commonalities with mast cells. Why? In their specific granules, they contain histamine and heparin. And you know that histamine is a very potent vasodilator, and inflammation mediator, and they can cause inflammation process when there is infection. And with these they all these granulocytes, these three type of granulocytes, they are phagocytic cells, these granulocytes. And the largest population is neutrophil, about 60 to 70%. Eosinophils, neutrophils appear due to bacterial infection. Eosinophils due to helminthing or worm parasitic infection. And basophils, which are less than 1%, they are very potent mediators, inflammation mediators, and they contain histamine, which is very important vasodilator, and they can cause anaphylactic shock. And these two cell types, mast cells, which are connective tissue cells, and basophils, they can cause such anaphylactic shock by secondary exposure to the antigen or allergen, for example. Yes, when secondarily these cell types, they both contain histamine. They also have receptors for immunoglobulin E, both these cells. And immunoglobulin E can attach to the surface of basophils and the mast cells. And after antigen appears and binds with is this immunoglobulin A, the degranulation of all these granules takes place, and the blood all over the whole body, and the blood pressure may drop, and this is very lethal condition. Now, about mast cells. Mast cells, they are round cells. They have a lot similarities with the basophils, and they first in vitro differentiation and growth of a poor population of mouse mast cells revealed that these cells need interleukin 3 for their differentiation, and this interleukin 3 is released from T lymphocytes, and they are present in connective tissue. They are in vicinity of blood vessels. They are called perivascular mast cells, and their differentiation depends on T cell. Then they are found in connective tissue, in skin, mucosa, lungs, digestive tract, mouth, and conjunctiva, and the nose. And they play key role in inflammatory process, and they can, after activation, and they can selectively release histamine. Now, the next cell type is, what is it? Yes, these are defensive cells. These are neutrophils. These are leukocytes, granulocytes, and you can see neutrophil, eosinophil, and basophil. How to recognize neutrophil? Neutrophils, they have such segmented nucleus, such segmented nucleus, these lobes, lobulated or segmented nucleus, and these lobes are from 2 to 5 lobes, and here you can see this neutrophil, and it is very easy to recognize. And how to recognize eosinophils? Eosinophils, when their count is increased, it may indicate they are modulators they modulate the allergic reactions. And how to recognize them in blood, on blood smears? In blood smears, you can see that they have granules, and these granules are pink or red, and they have a bilobed nucleus. And the third type, basophil, which contains heparin and histamine, you can see here it is very, it has abundant these granules which obscure the nucleus, and the nucleus also is bilobed. And here you can see this is the eosinophil. They are also found in normal pulp, and they increase during inflammation, eosinophil. And here you can see that these are the nucleus consists of two lobes, and these two lobes are connected with very narrow braid of chromatin. And here you can see this is increased cellularity of pulp. In pulp infection show larger number of cells, and you can recognize the plasma cells, lymphocytes. You can see the neutrophils and fibroblasts. You can see here, and you lymphocytes, these very dark and compact cells, and also you can see here plasma cells. Then uh, uh, yes, these are lymphocytes. Maybe there is the defensive cells. What cell types? Yes, we I have to say some words about dendritic cells. They are these dendritic cells, also are, they are antigen presenting cells. and they act as messengers between innate and adaptive immune system. They have such sheet-like. We have not another. Yes, they have dendritic cells. They have sheet-like processes that fold into the membrane surface, and the dendritic cells are present in those tissues that are in contact with the external environment, such as skin, for example, in the inner lining of nose, lungs, oral cavity, stomach, intestines. They can be found in the immature form in the blood, and once activated, they migrate to the lymph nodes where they interact with the T and B cells to initiate adaptive or specific immune response. At certain stage, they develop branched projections, the dendrites, such projections, large projections, and dendritic cells are derived from hematopoietic bone marrow progenitor cells, and these cells initially transform into immature dendritic cell, which constantly search surrounding environment for pathogens such as viruses or bacteria, and this is done through recognition receptors, pattern recognition receptors, and on their surface. And they also are phagocytic cells, and they can engulf and digest as macrophages, and after contacting with antigens, they become activated and migrate to the lymph nodes. In non-lymphoid organs, macrophages and the B cells can only activate memory T cells. But whereas these dendritic cells, they are very professional antigen-presenting cells, can activate both memory and the naive T cells, which is very important process in acquired immune response. And there are also lymphocytes. You know that the lymphocytes, they are very important cells, T and B lymphocytes, and B lymphocytes, they have antibodies, gamma globulins on their surface like receptors. But T lymphocytes cannot bind carbohydrates or other than protein receptors such as MHC complexes. Please keep in mind that T lymphocytes only recognize remnants, reminder of pathogen on the MHC complex, which is called major histocompatibility complex. And after recognition, it activates and releases some interleukins, for example, interleukin 4, which can activate B cell. And after activation of B cell, here you can see, after activation of B cell undergoes the clonal selection, undergoes gene clonal selection, and it converts into the such cell which is called plasma cell. Plasma cell is mini factory for antibody production. These plasma cells, which are derived from B lymphocytes, can produce antibodies. And you can see that nucleus, which is acentrically and shows nuclear chromatin condensations at the periphery. And also you can see the plasma cell cytoplasm, and it contains a large amount of rough endoplasmic reticulum and Golgi complexes. And here near this cell, you can see the macrophage. And here, this is the how to recognize plasma cell in blood smear. You can see that it has asymmetrically located nucleus and a large plasma cell. You know that this, you can see this lymphocyte. Lymphocyte has a very small rim, scant rim of cytoplasm around the nucleus. Nucleus is centrally located. It has very small cytoplasm and nucleus is very condensed. If you compare to this plasma cell, you can see that, and here plasma cell, it has such fried egg appearance, and it has increased cytoplasm, and this increased cytoplasm is at the expense of increasing of endoplasmic reticulum and Golgi complexes. Then, what is it? A graph of lymphocyte. Yes, you can see lymphocyte and centrally located nucleus, and this nucleus located in pulp, and the cytoplasm forms narrow rim. Here you can see the narrow rim around the nucleus, and nucleus is dense and condensed. And here you can see the dendritic cell present in odontogenic zone of the pulp. Then, what is this and what is orthochromatic mast cells? Yes, this is mast cells of calcium carbonate phosphate abundant in odontoblastic layer. And here you can see mast cells. They play a role in inflammation. They contain, as you can see that yes, they have such specific feature as it is metachromasia. What does it mean? They are granules by their concentration. Maybe they can differently stain these granules, and it gives such different color granules, such as from purple to deep blue, and this process is called metachromasia, and it contains histamine, which is the mediator of inflammation and help defend against pathogens, and are involved in wound healing and tissue repair processes, and they can detect and respond to foreign substances, and when it is activated by a trigger, these granules release many mediators, chemicals such as these mediators of inflammation, and they are such bridge between innate and non-specific and specific immunity. And here this is electromicrograph of these cells, mast cells, and you can see the exaggerated granules, specific granules with this mediator of inflammation. Then, after this, yes, we can now discuss the yes, we can discuss the circulation and vasculature blood vessels. You know that pulp is extensively vascularized, profusely vascularized, and it has such blood supply. And pulp, here you can see, this is pulp, vascular cast of the mandibular molar, and you can see blood vessels. The main vessels pass in and out of the pulpal tissue through an apical foramen. Blood vessels also pass through the accessory canals, and these lateral vessels often contribute more to the blood supply of the pulp than those entering through the apical foramen. Via the numerous lateral canals, the pulp is also connected to the blood vessels of periodontal ligament, and the vessels that enter the root pulp are small, narrow arteries and thin-walled arterioles. They enter to tooth as a bundle and initially increase right in the middle of the pulp and along the canal walls toward the coronal pulp where they branch markedly. And here you can see this branching. Arterioles are arterioles in circulation, in the over the body. Arterioles are small blood vessels, arterioles. They have about two or three smooth muscle layers around the lumen, these arterioles, and they are called resistance vessels with a diameter of approximately 50 micrometers. And these several layers of smooth musculature regulate vessels tone. And arterioles, why arterioles are very important in blood supply? Because they feed, these arterioles feed capillary beds. And capillary beds, which consist of 10 to 100 capillaries, very small capillaries, where gas and nutrient exchange take place, are, are supplied by these arterioles. The transitional region between arterioles and capillaries is known as terminal arterioles or metarteriole. This metarteriole is small branch of arteriole which has both arterial and capillary capillaries functions. And although it has the same circumference as the capillaries, it is surrounded by more musculature. It has such musculature bundle, smooth muscle rings or sphincters, and by contraction and relaxation of these sphincters, it regulates blood flow to the capillaries, to the capillary beds. Yes, so the capillaries then have an internal diameter, the small capillaries, about only 8 micrometer, which roughly corresponds to the diameter. You know that RBCs or erythrocytes, they have the standard diameter from say 7 to 8 micrometers. And these sphincters of smooth musculature are interposed between the terminal arterioles, and the capillaries are under neural control because autonomic or sympathetic nerve endings are exist here in these walls of these arterioles, and the cells from the surroundings can also have an effect on the sphincters and hence on blood flow in these capillaries and supply of capillary beds. Uh, and this makes it possible to produce a variable blood flow in highly localized regions of the pulp, and inflammation can hence be locally confined without influencing the entire pulp. Then let's see this picture. Mhm. And you can see that this is the capillary loops, the vessels, they divide and narrow to some degree in the root canal, but branch profusely once they are within the coronal pulp. Here you can see coronal pulp, and capillary loops extends towards the dentin, and this looping is mostly easily demonstrated in animal preparation because they can be perfused through the vascular system and caused prepared. Here they see this branching. Then mm, yes, This is there are three layers of the, yes, there are three layers of terminal supply to the odontoblast layer. Most superficially, you can see they are flat-ended capillary loops, these flat-ended capillary loops. And there is then after, beneath this is the capillary network, and under you can see the venular network. There are straight arterioles and their branches which supply this capillary bed. The capillary network beneath the odontoblast is dense enough to be known as subodontoblastic capillary plexus. And here we can see this is capillary plexus beneath the odontoblastic layer. And here you can see these are pulp capillaries, which are very close to the odontoblasts. And here you can see these are capillaries, which are very near to odontoblast layer. And there, the capillaries in the this subodontoblastic zone are fenestrated. Here you can see these are the capillaries. They are fenestrated. And what does it mean? You know that there are three types of capillaries. There are continuous capillaries, which are in muscles, in brains, and they have the continuous layer of endothelial cells and continuous basement membrane. The second type of capillaries are fenestrated capillaries, which have the fenestrations or small holes in the wall of endothelial lining, but they have continuous basement membrane. And such capillaries, fenestrated capillaries, are found in this layer, in the subodontoblastic layers. Then these fenestrations are 60, 80 nanometer in diameter. These fenestrations are small holes, and only basement membrane is present at the fenestrations. And this allows rapid movement of materials out of the capillaries. Mm. And this the non-fenestrated capillaries leave open a gap of only 5 to 10 nanometers between the individual endothelial cells, which is held together by gap junctions, because you know that in a non-fenestrated continuous capillaries, the endothelial cells are overlapping each other, and sometimes the fluid from plasma leaks from these capillaries. But these gaps are they are very small, these gaps, and fenestrated capillaries, they facilitate the movement of fluid out and into the capillaries. Yes. These non-fenestrated capillaries are found during early dentinogenesis in the vicinity of young odontoblasts. And when dentinogenesis in full progress, the capillaries become fenestrated, and these fenestrations measure approximately, as we mentioned, 60 to 80 nanometers. The blood vessels from a dense subodontoblastic microcirculation plexus, which can be divided into three layers. The first is known the first is known as the odontoblastic layer. The second layer, also known as the capillary network, lies just below it and contains convoluted precapillary and postcapillary sphincters, and the network of arterioles and venules run in the third layer of the very bottom. The venules and these capillary loops merge into the collecting venules. After capillary beds, these capillaries unite and form the venules. And these post-capillary venules are very important in inflammation because these are the sites from which the granulocytes and the blood-formed element, phagocytic elements, leave the circulation. And the venules and the large veins which have wider lumens than the arteries extend into the center of the coronal pulp, and run along the arteries to the apical foramen where the veins emerge from the pulp. Veins also leave the pulp by the lateral canal. Uh, and blood vessels with the thicker cross section run in the root pulp, whereas a highly branched network of finer peripheral vessels lies in the coronal pulp. And the blood vessels in the pulp, is known as microcirculation. And there are also lymphatic vessels. You know that this blood circulation causes in all capillaries hydrostatic pressure. Hydrostatic pressure is the pressure from fluid which is exerted on the walls of the blood vessels, and these capillaries they leak, and the interstitial fluid is formed, and the drainage of this tissue is by lymphatic capillaries and lymphatic system. And the lymphatic capillaries, they are such close-ended capillaries. They are formed by these endothelial cells, which are lymphatic endothelial cells. It is no difference between, it is very hard to differentiate blood vessels endothelial cells or lymphatic endothelial cells, because they have the same markers, they have common markers, similar markers, and they form such small lymphatics which have such openings or micro valves on their walls, and it provides the unidirectional movement from interstitial fluid into the such blood lymph vessels. And it is very difficult to differentiate these lymphatics and to differentiate lymphatic. Here is supportive element. Lymphatics, to differentiate lymphatics from blood vessels, blood capillaries, they have very thinner endothelial wall, which has smooth musculature but no basement membrane. Lymph vessels bear vessel valves. The larger lymphatic vessels, they have lymph valves, which are the protrusions or folds, endothelial folds, and they are similar to those in larger veins. And the red blood cells are never, how to distinguish veins which also have such irregular lumens such as lymphatics, how to distinguish these blood vessels from veins. They never contain, in normal condition, blood cells inside it. Uh, yes, what is it? Supportive lymphatics. Yes, vessels have been identified in the pulp at the ultrastructural histological levels. Yes, by the answers. And the lymph from the dental pulp collects in the submaxillary and submandibular lymph nodes, from which it pours into the lymph nodes network of the superficial jugular plexus and the deep jugular plexus, both of which open into the jugular trunk. Now you see you know that we have mentioned that the dental pulp is very well innervated, and there are different types of innervation, and this innervation takes place from this trigeminal ganglion. You know this trigeminal ganglion already, and yeah, the several large nerves enter the apical foramen of each molar and premolar, with a single ones entering the anterior teeth. A young premolar may have as many as 700 myelinated and 2,000 unmyelinated axons entering the apex, and these nerves have two primary modalities, and we discuss it very soon. And you here you can see the distribution of nerves from trigeminal ganglion. As we mentioned previously, was mentioned, the dental bulb is one of the most densely innervated tissue in the body. Yes, yet sensations and the mild pain are rarely registered from the pulp, and the several pain is perceived more intensely. And here we can, yes, and nerves enter the pulp through the apical foramen, you can see, and lateral canals, depending on size, axon diameter, myelinization, or conduction modality. There are six morphological distinct types of nerve fibers, and there are unmyelinated nerve fibers, largely belong to the autonomic nervous system. These are mainly sympathetic autonomic fibers. They are found in pulp. They Most of these run along the blood vessels, innervating their musculature and thus regulating vasoconstriction and vasoconstriction. And you know that this causes the pulsatile vasoconstriction of this terminal arteriole or metarteriole, and this causes the distribution of blood through the blood to the capillary beds. But there are also afferent sensory fibers. They arise from the maxillary and mandibular branches of the fifth cranial nerve, trigeminal nerve, and these myelinated nerve fibers belong to the trigeminal nerve. They are somatosensory afferent neural pathway that have free nerve endings and transmit diffuse but intense pain. On entering the root pulp, the axons are combined into two or three nerve bundles. They possess they possess myelinated sheath, but by the time at the to the coronal pulp, they lose this myelinated sheath, and approximately 150 but maybe 1,000 and 500 individual axons run within these nerve bundles, and the average diameter is about 4 micrometers, and these bundles of nerve fibers mostly remain combined as they pass through the radicular pulp. Once the myelinated sensory nerves reach the periphery of the coronal pulp, they lose their myelin sheath and are merely surrounded by the Schwann cells and the basal lamina. Thereafter, they can no longer be distinguished structurally for the unmyelinated fibers of the autonomic nerve system. In the bipolar tissue zone, here you can see this cells bridge zone or bipolar tissue zone. uh, this cell, these nerve endings, nerve fibers branch repeatedly and form the Rashkov plexus. Rashkov plexus, you can see here in a yellow Rashkov plexus. And this nerve plexus is not formed until eruption and takes on its final form after eruption is completed. When the tooth root is fully developed after eruption is completed and development of the primary dentin is completed, the Rashkov plexus begins to be shaped under the pulp horns from which it extends in the direction of the root pulp. The finished Rashkov plexus consists of bundles of unmyelinated axons, each of which is surrounded by a Schwann cells and its basal lamina. Small bundles of two axons and individual axons without Schwann cells are also found in the cell-free rails zone. And such free exons can barely be distinguished from unmyelinated cells, and the terminal branches of the nerve plexus of Rashkov pass through the row of odontoblasts, and we see it here, and this is plexus of Rashkov, and they can pass between these odontoblasts, and sometimes they can extend into the dentinal tubule, and here they are no longer surrounded by a Schwann cell, and these nerve endings run for at least 0.2 millimeter in the dentinal tubules, but can also reach as far as the dentino-enamel junction, and where they exist in no dentin canals as free nerve endings. Innervation of the dentinal tubules is particularly intense in the area of the pulp horns. On the side walls of the crown and toward the root, fewer dentinal tubules are permeated by the nerve fibers. Hello, are you here? Are you tired?
A little bit, doctor.
Yes, yes, now, very, very soon. Let's discuss and we finish this topic. Then you were so quiet that I think that you are asleep.
No, I'm not sleeping.
Yes, yes.
We
Yes,
are
you
here.
can see this is the Rashkov plexus of Rashkov, and you can see its branches which approach these the dentin, predentin, and dentinal tubule. And here you can see this is immunocytochemical section, which shows this is the calcitonin gene peptide, receptor peptide, and you can see this is special staining for this growth factor, and fibers you can see branching extensively in the coronal pulp and entering into dentin for up 1 millimeter, but avoiding reparative dentin. And here you can see reparative dentin, and maybe we can see this plexus of Rashko, and you can see the branches which approach they are through the odontoblast. This is odontoblast and even odontoblast processes and tubules. The nerve endings do not have any connection with the odontoblasts via either tight or gap junction or synapses. They have no junctions, these nerve endings, they have no junctions with the odontoblasts. In the area of interradicular and tertiary dentin, innervation is of particularly low density, as you can see here. At the tertiary dentin is low density and maybe entirely absent innervation. The pulp of the anterior teeth in the mandible can be reached by branches of the mylohyoid nerve, which runs lingually in the region of the mandibular symphysis. The role of the dentinal tubules in conduction of this, you know, that dentinal tubules, they contain a fluid which is called the tubular lymph, and this composition of this lymph, ion composition of this lymph, potassium ion and sodium ion composition can trigger and generate electrical impulse. And the signals in the nerves are conducted in the form of electrical action potentials, you know. This happens within seconds or shorter time units, and these neuropeptides are conducted by the nerves from the pulp in the axons to the neurons in the trigeminal ganglion and back to the pulp. And now about these axons, about the type of these fibers, you know that there are six types of fibers. They are A beta fibers, have a diameter from 5 to 15 micrometer, and action potential conducting speed is about 30 or to 70 milliseconds, and they make a small proportion of the fibers, and they are medium sized and myelinated, and are primarily found in the dentin pulp junction. Here you can see these are fibers, and dentin pulp junction and the pulp horns up to cusps, and they innervate the dentin as they also run in the dentinal tubules. Then, as a result, there are extensive contact zones between odontoblasts and nerve endings at the A beta fibers, and there are also A delta fibers. They have a diameter from 1 to 7 micrometer and are divided based on conduction speed into the fast and slow fibers. And the slow fibers, the A delta slow fibers, are polymodal fibers, and they account to 20% to 50% of the fibers. And the last A delta fibers are thin and myelinated and reach the odontoblasts, the predentin and the dentin, and extend into the dentinal tubules and establish contact with odontoblasts. Their ends lie very closely adjacent to the odontoblasts, but there are no gap junctions or synapses between them. And also a paracrine signal exchange would nevertheless be possible because of the close proximity. You know, the paracrine is when the cell releases the factor, growth factor, or other signaling molecules, and it's in around close proximity, only very close on very close located cells. And also there are the slow A delta fibers. They are polymodal, have a conduction speed from 2 to 10 milliseconds, and they reach the odontoblasts and fibroblasts, and they do not penetrate the dentinal tubules. And these fibers, they convey the impression of a localized sharp pain. And also there are C fibers, which are unmyelinated and conducted dull, difficult to localize pain, and their diameter is 0.3 to 103 micrometer, and the action potential conduction speed is 0.5 to 0.05 milliseconds. And this majority of these fibers are unmyelinated in the teeth, and on entering the tooth, their axon diameter is approximately 10 micrometers, and they get thinner because of the more extensive branching in the cervical region of the loop, and in the coronal area they are all unmyelinated, and they reach the cell-rich zone and the cell-free veil zone and the odontoblast zone. Many of these fibers and along the blood vessels. And here you can see this is the immunohistochemical method. They demonstrate the nerve endings which surround. Here you can see they surround the blood vessels. These are nerve fibers, and in the odontoblast layer they reach odontoblast layer. The distribution of the nerves is essentially the same in primary and permanent teeth. The fibers of the sympathetic innervation system are less numerous than the sensory fibers. They are found primarily in the center of the pulp tissue, and many nerve fibers contain neuropeptides and are able to discharge them, and also fibroblasts, they can produce the neurotrophic factors in response to injury. Expression of these factors is altered, and these alterations are perceived by surrounding odontoblast immunoactive cells and the nerves with their receptors, which have high affinity to these neurotropic factors. Then these cells, all these cells in dental pulp, they can respond instantly to endocrine, paracrine, and autocrine signals. And you know that the nerves cannot do this instantly. Their cell nucleus, which controls the functioning of the cells, lies in the trigeminal ganglion for the sensory neurons, up to 10 centimeters away, and the neurons of sympathetic fibers lie in the cervical sympathetic ganglia, and for the proportion of the signals exchange that is not based on electrical principles but on the flow of signaling molecules. And this is proteins. It is very important, necessary for the signaling molecules to be transported out of the pulp tissue in a retrograde direction. And the one moment I'd like to mention is the uh, yes, you know that nociceptors, and we say that nociceptors are pain receptors. Uh, nociceptors, pain, and nociception are not synonyms. Let's discuss this question. Pain is a subjective sensation, is defined as an unpleasant sensory and emotional experience associated with actual or potential tissue damage, or described in terms of such damage. Nociception, on the other hand, describes a series of subjective neuronal processes, and is defined as the reception, conduction, and the central processing of noxious signals. And these noxious signals may be these signals and signaling molecules. Nociceptors are essentially receptors that respond to harmful or potentially harmful stimuli. Pain clearly can be a result of stimulation of nociceptors, but so are some reflexes and changes in arousal state without the organism sensing pain. Nociceptors are thought to be free nerve endings and appear to respond specifically to noxious heat, intense pressure, or irritant chemicals, but not by innoxious stimuli such as warming, cooling, or light touch. Fibers innervated the region of the head arise from cell bodies in the trigeminal ganglion, and there are considered to be three major classes of nociceptors. These are thermal, mechanical, or polymodal. And the thermal nociceptors, which are activated by extreme temperature, less than 5 degree and more than 45, are innervated by small myelinated A delta fibers that conduct impulses about 5 to 30 milliseconds. And there are also mechanical nociceptors, which are activated by intense pressure applied to the tissue in which they lie, and are also innervated by A delta fibers. And polymodal nociceptors, they are activated by high intensity mechanical, chemical, or thermal stimuli, and are innervated by either I delta fibers or are myelinated C fibers or autonomic fibers. And one question I'd like to discuss here, you can see these are unmyelinated nerve endings, and here you can see the Schwann cell and its nerve endings. They embracing in they are inside the Schwann cells, which wrap these nerve endings. And here you can see the distribution of, this is immunohistochemically for CGRP, this is calcitonin gene receptor peptide. And you can see this, the tooth in which cervical cavity was earlier, and this is normal control tooth, and there is the this is normal, and this is prepared cervical cavity. And there are age changes in dental pulp. We mentioned it. There are during the course of lifelong dentinogenesis, dentin is not always deposited layer by layer on inside walls, and there are sometimes they arise the accumulation or rounded structures known as denticles. And here you can see which are pulp denticles or pulp stones, called pulp stones. True denticle, there are two types of denticles: true denticle and false denticle. True denticles, they can contain such tubules. They are constructed as dentin by odontoblasts, and they have show dentinal tubules. And the false, they are mineralized connective tissue, calcified connective tissue. And these denticles may lie freely in pulp tissue or protrude out of the, and we can see here, you can see these denticles. These are nerve endings. Yes, you can see these denticles, these pulp stones here inside the root pulp. And here you can see this is false denticle. The center of the stone is lighter than peripheral. That indicates that it can contains the cell inclusions, and the pulpitis is present in the periphery of pulp chamber and the inflammation processes. And also you can see that the some physiological processes takes place. You can see it is attached to the, this is pulp stone which is attached to the dentin, and also this is free pulp stone which is embedded into the pulp connective tissue. And here it is denticles, false denticles. And what is age changings? And yes, there are age changes, which is the physiologic changes. You know that the throughout life dentin is laid down, and secondary dentin, and secondary formation of secondary dentin, it decreases the pulp cavity. And sometimes this pulp cavity is almost entirely obstacled. And the functions, yes, what is the functions of pulp? And here, what is it? Severe acute inflammation of pulp, it is root canal. And acute inflammatory cells, you can see there are these cells of immune defense cells. And here you can see the diffuse calcification appears as irregular calcific deposits in the pulp tissue, usually following collagenous fiber bundles of blood vessels. And also you can find fibrosis with the age of the pulp. Fiber content increases and cellular content decreases, and this is fibrosis when fiber bundles arranged in random diffuse fashion. And here let's speak about the physiology and the functions of the pulp. You know that pulp provides nutrition for the odontoblasts. First of all, odontoblasts are the superficial layer of pulp and they are derived from pulp. And pulp provides nutrition for odontoblasts. Mm, yes, and also it has inductive. It causes the, you know, that there are undifferentiated stem cells. Their differentiation gives the pulp is reservoir of cellular population and the repair processes. You know that this is associated with the network of unmyelinated nerve fibers and the subodontoblastic capillary plexus, and they give nutrition for these differentiated cells of all type of cells. And also you can know that the pulp has formative. It produces the dentin formation. This it was formation of primary dentin, secondary dentin. This formation of these two types of dentin is physiologic process, and also it gives rise to odontoblast-like cells, which are mesenchymal stem cells, and they can produce tertiary dentin, reparative dentin, and it has nutritive dental pulp maintains vitality of dentin by providing oxygen and nutrients to odontoblasts, and also it has protective function. We discussed it a lot of, there are immune system defense cells, and these defense cells defense or the reparative cells, defense cells, which are immune cells, they provide defense mechanism for pulp. Thank you very much. This is not included for tomorrow. Yes, of course, this is not included for tomorrow. For tomorrow, uh, four topics. Listen, please. Four topics, which are tooth morphology. There are questions from this tooth morphology. You have MCQs, and the questions are similar or the same. From vasculature and innervation, some questions, only some questions, enamel and dentin. You have all questions, all MCQs or questionnaires, and you try to answer, and don't worry. Any questions? Yes.
Only we should resolve the MCQ question
Which
on the
question?
drive. MCQ question on the drive, yes?
Yes, that questions, yes, and similar. Some questions I put such critical thinking questions. You must think and answer. And these questions from email, dentin, similar, very, I think, very easy questions. You have such questions, and you can answer.
Okay,
The
thank you very much,
appearance of oral
doctor.
cavity, dento also structure. There are questions, tooth morphology, and some questions may be about maxilla and the mandible. Only these four topics.
Okay, thank you very much, doctor.
Mhm.
Sorry, you can tell us from which is the title of the slides only for
The
this
of
topic
this, yes, I can. Yes, let me.
because I'm
Hmm.
confounded by him.
Yes. Dental pulp.
Okay, 1 second. Dental
Mhm.
pulp only?
Yes, yes, dental pulp. Yes. We discussed dental pulp. It was very large lecture because the topic was very large.
Yes.
Yes.
That
Yes.
yes.
Have you any question
No, thank you. Dental pulp only for today, yes?
for today? Not for a
Yes.
midterm.
Okay,
It
okay.
is. It will be on the next midterm. Midterm
Okay.
two.
Yes.
Yes. Then, if you have no more questions, goodbye and good luck.
Thank you. Thank you, doctor. Thank you.
Thank you. Goodbye. Bye bye.
Bye.